To go further on the structure and function relationship of different proteins, we already learned the structure of proteins. How the structure of proteins identifies or changes the function of a protein will take into consideration an important protein, and that is collagen and elastin in this session. Collagen is one of the most abundant proteins that is present in the body. It is one of the most abundant connective tissue. It is made up of 3 polypeptide chains. I am giving an overview of the structure. Each chain is approximately nearly 1000 amino acids. Each chain is called as an alpha chain. Now, depending on the amino acid sequence difference, there are different types of alpha chains like alpha 1, alpha 2, etc. Collagen is made up of 3 main amino acids. It is always represented by the formula easily as glycine X, Y. What can be X? What can be Y? X can be proline and Y can be hydroxyproline or hydroxylysine. We'll go further with the synthesis of collagen. Where they are synthesized, how they are synthesized, what are the things that are happening during the synthesis of collagen. Now, collagen, the cells which are responsible for the synthesis of collagen are the fibroblasts, the osteoblasts, the chondroblasts. All these cells can synthesize collagen. So what do they do after they synthesize collagen? It is secreted into the extracellular matrix. Now, in collagen, there is a lot of hydroxylation and glycosylation that is taking place. These type of modifications which take place after a protein is synthesized is called as post-translational modification. To give an overview of what exactly is the structure of collagen, we call it as a triple helical structure. The 3 alpha chains of collagen, as I said, it is triple. There are 3 chains. The 3 alpha chains of collagen, they are helically, they are not just one helix, but there are 3 types of them. Each of the chains are woven around each other and they form a different structure, and this is called as the triple helical structure. These 3 alpha chains which are intervened together, this is called as a tropocollagen. I'll go further. I'll tell you as the synthesis is taking place, how it is synthesized, but giving an overview. This is called as a tropocollagen. Now, many such tropocollagen molecules, I'll just show it briefly as this. Many such tropocollagen molecules, what happens is all these will aggregate together. This type of arrangement in which the tropocollagens are arranged, this type of structure is called as a quarter-staggered structure. This whole thing, this type of quarter structure, each of these is like this, triple helical. This quarter is called as a collagen fibril. This is called as a collagen fibril, which has many tropocollagens and arranged in the form of a quarter staggered. Now, there is a reason why you want to know about this is there is a disease which can affect collagen synthesis, and that is the Ehlers-Danlos syndrome. In this, the fibrillar collagen is affected and it leads to vascular problems, fragile stretchy skins and loose joints. Now, to go further on the synthesis of collagen. Now, synthesis of collagen. What is it? It is a protein. So, where it has to start, how it takes place, and how the structure contributes to the function. Now, it is a protein. So, the synthesis of this is coded by the chromosome. So, the synthesis of it will start in the nucleus. So, in the nucleus, we have the DNA. In the DNA, we have the genes for collagen. So, these genes, the mRNA is transcribed. This mRNA will come out of the nucleus. Now what happens? It has to get translated. DNA transcription to mRNA has to undergo translation. So for this translation, different ribosomes will come and attach to the mRNA. Different ribosomes will come and attach. Once the ribosomes attach, what will happen? The ribosomes will process this mRNA and it will result in the formation of the first few amino acids, and this is called as this with the amino acid chain, this is called as the pre-pro alpha chain. Pre-pro alpha chain. Now this pre-pro alpha chain has got a signal sequence. This signal sequence tells it where to go. So it goes into the endoplasmic reticulum. It is in the endoplasmic reticulum that further changes takes place of this alpha chain. Once it goes there, minute it goes there, the signal sequence of this pre-pro alpha chain, whatever signal sequence it has got, which told it its address where to go, this signal sequence is cleaved off. First thing that happens is the signal sequence is cleaved. Now what happens? This pre-pro alpha chain is now converted into pro alpha chain. This is an alpha chain. Remember, it is not the same as the alpha helix of the secondary structure. This is still a chain. There is no helical formation. Now this is the pro alpha chain which has been formed. Now what happens? Certain enzymes will act on the pro alpha chain and it results in the hydroxylation of certain proline and lysine residues on this. The pro alpha chain gets hydroxylated. It is still a pro alpha chain but is hydroxylated. What has been hydroxylated? The proline residue has been hydroxylated and the lysine residue has been hydroxylated. I'll give the reaction in detail later. Now for this hydroxylation, most important thing that is required is vitamin C. Ascorbic acid is required, molecular oxygen is required. All this will cause hydroxylation. So first change that has happened, this is a post-translational modification and that has happened is hydroxylation. The second change that is going to happen to this is these hydroxylated, some of the hydroxylated residues of especially lysine are going to get glycosylated. So I'm going to show this as a glycosylated. Some residues are going to get glycosylated. This is also a type of this hydroxylation, and glycosylation is a type of post-translational modification. Again, which are the substances which can be glycosylated include glucose or galactose. So glycosylation takes place. Now this whole thing which has been glycosylated, which has been hydroxylated, this is the pro-alpha chain which is under, this is still a pro-alpha chain. Now what happens? It has to get assembled together. So what happens is many such pro-alpha chains, they all come together. These pro-alpha chains are going to come together. What will happen is disulfide bridges are going to be formed between them. Disulfide bridges. The minute the disulfide bridges form between the chains, it helps the chains to come together. Now this is called as a pro-collagen. Pro-collagen is where the pro-alpha chains have come together. We call it a pro-collagen. This whole thing is taking place in the endoplasmic reticulum. This is happening in the endoplasmic reticulum. Now what has to happen? This enters into the Golgi. Remember, from endoplasmic reticulum, it goes into the Golgi. I'll talk about this. I'll just rub the board and now I'll tell you what happens in the Golgi. The pro-collagen is now in the Golgi apparatus. This Golgi, now what it does, this pro-collagen, it gets secretes. The function of the Golgi is to form secretory vacuoles. So it forms secretory vacuoles. And what does this secretory vacuole contain? It contains pro-collagen. So what is a pro-collagen? The pro-collagen is the pro-alpha chains which are linked by disulfide bridges between intrachain disulfide. It still has a bit of a helical structure, but it has some extra portions to it. So this pro-collagen which is present within the vacuole is now secreted out of the cell. this comes out. This comes into the extracellular matrix. This is still linked by disulfide bridges. At the N terminal or the carboxy terminal, in both places, it is linked by the disulfide bridges. This pro-collagen molecule, which has now entered into the extracellular matrix, it will be acted upon by certain peptidases. These peptidases are going to break off at both the ends, the N terminal, wherever the disulfide bonds are there, or the carboxy terminal, where the disulfide bonds are there. Both these are going to be cleaved off. They are peptidases, they cleave it off. When this is cleaved, what is remaining now? Now this is a, what do you think it is? It is a tropocollagen. As I said, it is still not collagen. It has to still form a quarter-striated structure. Only then it becomes a collagen fiber. So right now it is tropocollagen. Tropocollagen is formed. Tropocollagen is procollagen which has been cleaved off. The N terminal and the C terminals have been cleaved off. It is formed tropocollagen. The next step is the formation of the quarter-striated, this triple helical structure. Now actually I have shown it as 3 strands, but they are all intertwined together like this. These intertwined structures are going to come together. This is one tropocollagen, this is another tropocollagen, another tropocollagen, another tropocollagen. In between all these are formed. So what stabilizes these tropocollagens together? So something must happen here, isn't it? So that bonds can be formed between the different tropocollagen molecules. So the formation of this bond is not a disulfide bond. For this, it requires another enzyme. And this enzyme that is required here is called as lysyl oxidase. This lysyl oxidase is different from the lysyl hydroxylase, which was responsible for hydroxylation of the pro-alpha chains. Lysyl oxidase is in the extracellular, whereas it was in the endoplasmic reticulum. You had the prolyl hydroxylase and the lysyl hydroxylase, which were acting for the hydroxylation of the proline and the lysine residues. Whereas lysyl oxidase is responsible for the conversion of lysine to al-lysine, or hydroxylysine to lysine is converted to lysine, or hydroxylysine similarly is converted to hydroxy-al-lysine. Now this is the al-lysine of one and hydroxy-al-lysine of another tropocollagen which form the bond between them. And this results in the formation of the final collagen molecule, that is the collagen fibril. Now one important thing over here is this lysyl oxidase requires copper. It is a copper-containing enzyme. Just as the way vitamin C was required for the formation of collagen, procollagen, and hydroxylation and further post-translation modification, copper is required for the extracellular modification of tropocollagen and for the formation of the collagen molecule. So here I have this in the form of a flow chart. Look here. The DNA transcription results in the formation of mRNA. The ribosome translation converts pre-pro-alpha chain with the signal sequence, which goes to the endoplasmic reticulum. Signal sequence is cleaved, results in the formation of pro-alpha chain, hydroxylation, prolyl and lysyl hydroxylases. As I said, the vitamin C containing enzyme require an absolute requirement for vitamin C, glycosylation with glucose and galactose. 3 pro-alpha chain assemble to form procollagen with intrachain disulfide bridges, enters Golgi, secretory vacuoles, procollagen is released into the extracellular matrix, cleaved, and tropocollagen is formed, lysyl oxidase acts, and al-lysine, hydroxy-al-lysine formed, cross-linking occurs, and the collagen fibril is formed. So that is in brief the synthesis of collagen. Now we go ahead to one more connective tissue, and that is elastin. What is elastin and how is it also structure function relationship between different proteins? Elastin. I want to tell the elastin is not as complicated as collagen. Elastin is a connective tissue. It has rubber or elastic-like properties, and that is why the name elastin. Now, where is this present? Wherever a tissue has to expand. Where do you think tissue expands? It is present in tissues like lung. The lung has to expand when you inspire. It has to be present in large arteries like aorta because the aorta has to expand. So wherever expansion is required, that is where elastin is present. It is also present in elastic ligaments. So wherever expansion is required, that is where elastin is formed. Now what is important here is, here also elastin is synthesized as a tropoelastin which is converted to elastin. Difference between collagen and elastin is, though elastin is also equally rich in proline and lysine, it contains very little hydroxyproline and absolutely no hydroxylysine. So the main difference is between collagen and elastin is lack of hydroxylysine. Now this elastin is scaffolded. It is doesn't form a quarter-staggered structure. It is mainly scaffolded. It also is present in different layers. However, it is scaffolded by fibrillin and then it causes crosslinks within the chains. Now one important disease which is caused because of a deficiency or a problem with elastin is the Marfan syndrome, where there is an abnormal fibrillin. Now, another important disease which is associated with this connective tissue is what is called as the alpha-1 antitrypsin deficiency, alpha AAT. I'll be talking about this also, alpha-1 antitrypsin, alpha-1 antitrypsin. Now, what is this? What does the name suggest to you? Antitrypsin means something which is against trypsin. So why do we call it antitrypsin? Where is trypsin found? Trypsin is actually found in the digestive tract. But this antitrypsin can act as an against many enzymes which have trypsin-like properties. Now where is it released from? Alpha-1 antitrypsin, before we go where it is released from, what is actually happening in the lungs I'll talk about. Now in the lungs, there are a lot of neutrophils. These neutrophils, whenever they are activated or when they get degenerated, they release an enzyme called as neutrophil. Released from neutrophils, so they release an enzyme called neutrophil elastase. So what does the name suggest to you? Elastase. So it is going to degrade elastin. Now this will, if it degrades elastin, if elastin is degraded, what will happen? This is degraded by, elastin is degraded by elastase. So what will happen? Elastin is degraded by elastase. So what will happen? The lung will lose its power to expand. The lung will lose its elasticity. So it can lead to a condition called as emphysema. So in the body, neutrophils are normally activated. They are normally undergo degeneration and all that. So elasticity is constantly released. So to prevent, we cannot prevent neutrophil from releasing elastase. However, the body has a defense mechanism, and that is what alpha-1 antitrypsin. Alpha-1 antitrypsin prevents this elastase. It binds to elastase and prevents the degradation of elastin. So that is how elastin will remain as it is and the lungs are healthy. So what can happen in emphysema? In alpha-1 antitrypsin deficiency, if there is a genetic deficiency, alpha-1 antitrypsin is not there, neutrophil elastase will degrade elastin, leading to emphysema. What happens in smoking? Now, we all know that smoking can lead to certain lung disorders. So what is happening in this is, smoking has a direct effect on alpha-1 antitrypsin. What it does, it causes inactivation of certain methionine residues in AAT. So when it is inactivated, alpha-1 antitrypsin is inactivated by smoking, it will again lead to elastase, degrading elastin, leading to emphysema. So that is the importance of elastin in this thing. So with this we talk about the structure and function relationship of two important connective tissues, that is collagen and elastin. In the next session, I'll be talking about hemoglobin and myoglobin, the two important proteins and their structure and function relation. Thank you.